everyone. My name is Greg. I'm a postdoc working at Lauren Riesberg at the University of British Columbia. I'm going to tell you a bit about homoploid hybrid species in sunflower. So the sort of old view of hybridization, probably before any of our times, was that hybridization was not really that important. That hybrids were formed, but they were inviable or infertile, and they probably died and they were not important. We now know that this is not the case, that hybridization actually can ultimately result in introgression. So introgression is where you have two lineages, and then they hybridize. The hybrid offspring are going to backcross into one of the lineages, and then you have two species, but in some populations, or in all populations, you have genes from one species moving into another. And we now know that this introgression can be fairly common, that it's found in a lot of different cases when we look in the genome, from humans to different kinds of plants to birds. And I think what the most extreme form of introgression is, and what I'm really interested in, is in hybrid speciation. So hybrid speciation is when you have two species, they hybridize, they produce a third lineage that has a mixed ancestry, and is also reproductively isolated from both of its parents. So it's a whole new species. The system I'm working on is Helianthus, so this is the sunflower genus. The two main species are Helianthus annuus, the common sunflower, and Pedalaris, the prairie sunflower. They're both annual species. They have really big and repetitive genomes, and they diverged about 2 million years ago. They are broadly sympatric across most central USA, and importantly, they have ongoing hybridization. So these two parental species have hybridized at least 3 times to produce 3 independent hybrid species. These 3 hybrid species are Helianthus anomalus, Helianthus deserticula, and Helianthus paradoxus. They each grow in sort of unique environments that the parental species tend to not grow on. So in sand dunes, sand sheet, or salt marshes. And it's important to note that these are homoploid hybrid species, not allopolyploids. So they have no genome doubling, which makes it a little bit harder to form hybrid species because of the lack of genome doubling. So if you imagine how hybrid speciation occurs, you start off with 2 parental species. They have their own unique genomes. At start, you're going to have F1 hybrids. Then what happens is those F1 hybrids are going to intermate amongst each other or backcross to the parental species, and you're going to have this sorting of the blocks of parentage. So you have all these different possible combinations, and as time goes on, what's going to happen is that drift or selection is going to fix certain combinations. So ultimately you'll get something perhaps like this, where you have this part of the genome is always from parent 1, and then this part is always from parent 2. And what I'm interested in is figuring out what these blocks look like in these hybrid species. And that can tell us about how the hybrid speciation event occurred. If you have really large blocks, that indicates that there was perhaps very high selection during the hybrid speciation, or very low effective population size. And that's because to get these large blocks, you have to fix really fast. As time goes on too much, there's going to be a lot of recombination. It's going to break up these blocks into really small pieces. So you can imagine if you have big blocks, it'd be small population size, high selection, or small blocks, you have low selection or high effective population size. The data set I'm using to answer this question is an RNA-seq data set. I'm using RNA-seq, but I'm only using it for SNPs, so I'm not actually using expression data at all in this. I have samples from both the parental species in red and blue, and then the 3 different hybrid species here. And I'm working with about 100,000 SNPs, which is reasonably dense for a genome this size. So there are different ways to measure block sizes, and there's some very smart people doing very sophisticated methods using HMM models that I tried on this data set and did not work at all, for reasons that I will explain. So I went back and just went to a very simple sort of approach to this, where you look for fixed differences between the parents, and then you look for strings of single parentage in the hybrids. So you're saying, okay, this is from parent 1, oh, switch to parent 2. So here we can see these are the markers, and then this is sort of the ancestry actually in the genome. There's some problems with this in that you're limited by your marker density. So here we would probably identify 2 junctions where it switched here and it switched here, but the actual genome is much more broken up, and we just don't have enough markers to pick this up. You can also have problems if you have incorrect parentage assignment. So here we think this is from the blue parent, but it's actually from the red parent. And one reason for that is just because of the sample size in the parental species. We only have about a dozen individuals from each parental species, so it's possible that something we think is a fixed difference is not actually a fixed difference, and we are misassigning this. One of the solutions to this is to use sort of a simulation where we simulate different junction densities with our real genotype density and then measure that. And we also include an error rate based on our sample size for our parents. This is going to look something like this. So we simulate this one junction here, we have our markers on this, and we infer 1. So we caught all the markers, all the junctions here. But then as we go up, we have more junctions. We miss some of them because we don't have enough markers, and you get another point here. And as you keep going, eventually you sort of level out where you just don't have enough markers to see any more of the junctions. Ultimately, you're going to produce some sort of a curve like this, and then you can take your observed junctions, so what happens in your real data, and sort of interpolate what the true junctions is based on your genotype density. This has been previously done using AFLPs and RAPDs for about 300 to 400 markers in the same parental species or in the same species, and they estimate about 0.5 to 1 junctions per centimorgan. So they're saying each block is about 1 centimorgan long, which is fairly large. My work shows a very, very different picture here. So if you follow this, it goes up to about 6,000. So the results are estimating about 5,000 to 10,000 junctions per centimorgan, dramatically more. And this is based on a lot more markers. It corresponds to a junction every about 300 to 500 base pairs, depending on the recombination rate. And that means we have a lot of examples of hybrid genes. So each of these rows is a single individual, and then the markers coded between the parents. And you can see that we have cases where it switches really fast here, where it goes from one parent back to the other parent. An important sort of sanity check on this, because it's so dramatically different than any of the previous estimates, is this less than random? So if you randomly assign your markers across the genome, you get a number that is higher than that. So the markers are actually grouped. It's not completely garbage. There's a caveat here, is that the current error estimates are based on sample size of the parents. So that is about 0.5% to 1%. And it's assuming that the parental species had modern allele frequencies when hybrid speciation occurred. And we know that this hybrid speciation occurred roughly several 100,000 years ago, and the parental species have gone through some amount of drift and also have interbred with each other. And so the parental species do not probably have the exact same allele frequency. I am having a little bit of a hard time figuring out exactly how to estimate this error rate based on probably some demographic models, but I haven't figured that out yet. And I just sort of tried, what if we just say, what if we're wrong a lot of the time? What if the error rate is 5 times or 10 times higher than I previously estimated? What happens to our scores then? Well, we get fewer junctions, so we get smaller or larger blocks, but they're still high. They're still in the order of thousands. So we don't actually get the same numbers that the previous estimates have, even if we assume that there's tons of errors happening in this data set. As I said in the beginning, there's 3 different hybrid species, so it's possible to look at the different hybrid species and see maybe some of them have more or larger or smaller blocks. And we can see some patterns here. So Paradoxus, one of the hybrid species, tends to have more junctions or smaller blocks, while Desertical and Anomalous tend to have slightly larger blocks. With the junction density, you can actually sort of use some calculations based on sort of neutral models for what's the effective population size that would produce this amount of junctions. And when you do the math, it works out to about 2,500 to 5,000 individuals. Previous estimates based on the really large blocks was that the effective population size was on the order of dozens. Well, this, I think, is a more reasonable number. The sunflowers, parental species, have extremely high effective population sizes on the order of about 1 million, and the hybrid species themselves have population sizes now of about 200,000. So starting off with this is, I think, not unreasonable. The question then becomes, what about selection? So we know that these 2 parental species have a lot of incompatibilities. These incompatibilities were almost certainly under selection during the hybrid speciation, and that selection should produce larger blocks. Well, the simulations I'm doing is a genome average. So it's trying to pick one number that fits the whole genome. So if there were some areas that had really large blocks and a lot of areas that had really, really small blocks, it's going to pick really, really small blocks because just how the simulations work. So just saying that in general, they're very small blocks. It doesn't mean that there aren't some larger regions in the genome. And some of the other work I've been doing has suggested that there are regions of the genome, rather large, that are predominantly from one parent or the other. So in conclusion, the effective population size during hybridization is probably much larger than previously thought in these two hybrid species, and that there is some variation between the different hybrid species, suggesting that there was differences in during the hybridization process. And I want to thank Lauren Riesberg, my supervisor, and the lab, and my funding sources. All right. Oh wait, yes.
I might have missed this. What is the presumed date of hybridization?
We haven't really done it with the new data, but it's probably on the order of like 300,000 to 500,000 years.
Okay. And have you done any work on the recombination rate and how that affects your blocks?
So the blocks are in centimorgans, right?
Oh, right.
Yeah. Although there are, so the hybrid species have had some chromosomal changes, and I'm not using their specific maps, so there are probably some differences in recombination rate that I'm not fully capturing. Yes?
I wonder if you can estimate the timing and mode of the introgressions with the distribution of the blocks.
Yeah, that's a really interesting idea.
Because your hybrid species seem to be formed at different times, or.
That's a good idea. We should talk later. That's good.